postdoc. And she was lucky enough to spend 3 field seasons in Ethiopia studying these wonderful monkeys. Her work resulted in some high impact and well-cited papers on vocal complexity. Morgan did her first postdoc at Princeton University, where she studied another species of monkey, marmosets. She worked on vocal development and biomechanics. Then she moved to the University of Texas at Austin, where she switched gears to working on smaller mammals, voles, and she studied both vocal communication and pair bonding. Morgan got bonded with voles in Texas, and when she moved in 2023 to Western, she brought the voles with her and kept doing research on the voles, and they have lived happily ever after, and Morgan will talk about them today.
Thank you so much. How's the sound? Thumbs up if it seems okay. All right, I see some thumbs down if it's not. Oh, you can't hear me, so too bad. Okay, thanks so much, and thank you also for having invited me for this wonderful day. I've really enjoyed meeting everybody and seeing all the different labs and all the different animals that people work with. I'm looking forward to, I think we have a few more interactions the rest of today, and definitely if you didn't get a chance to meet or if you want to follow up, I'm always happy to get in contact via email and have a chat. Zoom these days makes everything great. So feel free to reach out if there's anything you want to discuss or whatever it is. So let's go ahead and get into it. So like Rufin said, I'm bonded to prairie voles. These are the prairie voles right here. There's a male and a female. I don't know which one. I bet you guys can't tell either. These guys are very similar to each other from a phenotypic standpoint. Anyway, to kind of back up a little bit, I got into studying prairie voles because I had a strong interest in understanding social bonding as a phenomenon. And we know that what I mean by bonding is essentially the ability for an individual to form a selective preference for at least one other individual over others in their population. And a lot of species form these selective preferences or selective attachments. And so we see, for example, zebra finches form pair bonds between males and females. You see pair bonds between mating pairs in marmoset monkeys, in humans as well. And then you also see pair bonding outside of these male-female mating attachments. It can also be pairs between parents and offspring. You can think about even friendships. And so there's a lot of opportunities for there to be selective social partnerships in animal communities, both humans and others. But one thing I like to think about in terms of this social, sounds good.
There we go.
All right. Thank you. All right. Sound good? All right, amazing. Okay, so so widespread phenomenon. We know social bonding, social connections, and I'm very interested in thinking about social bonding from a longitudinal standpoint, both in terms of the lifespan of an individual, but also across evolutionary perspectives as well. And thinking about social bonding, not just from does it happen, does it not happen, but more so thinking about the complex dynamics that can happen throughout the process of forming these selective attachments. So you can think of social bonding as, at the very core, let's say involving two individuals, where you've got these communication signals or some sort of behaviors. These could be vocalizations, smells, visual cues, whatever it is. You've got these signals that happen in between individuals. And then you've got the side of more of the physiological perspective of how individuals have to produce but also perceive these signals from their social partners. So this is kind of the core fundamental aspect of social bonds. And then beyond this, then you can think about how these evolve across time. So on the one hand, you've got to pick potential partners. Then you've got to actually form some sort of selective attachment. And then beyond there, you've got to potentially interact with that same individual across time. And this can involve a lot of potential cognitive abilities as well, of recognizing a partner versus others, and having some sort of preference or reward that drives you to continually interacting with that other individual. So I'm very interested in understanding bonding from a variety of different perspectives, whether it be from evolutionary, developmental, and mechanistic perspectives, but also thinking about the timeline of how bonds are actually built, and the complexity there of kind of the early stages of forming a bond versus later stages. Before I get into kind of the nitty-gritty of the work, I do want to kind of back up, especially because I know there's a lot of trainees in the audience, and I know sometimes when I'm sitting in the audience, I always wonder kind of how people got to where they are. And so even though I don't have the opportunity to talk about everything that I've been involved in, I do kind of want to just touch on a few things, so that in case there's something here that you're like, oh, I really want to talk to her more about that, feel free to reach out to me. So when I went from undergrad to postdoctoral research, I started off actually in the University of Wisconsin at Madison, where I studied a little bit of mice. Didn't do a lot of mice work, but I mostly studied primates. So I worked with rhesus macaques and marmosets in the lab. I also worked in the field with howler monkeys and owl monkeys, which if you haven't heard of owl monkeys, look them up. They're these cute little owl clown-looking creatures that are monogamous and live in the trees in Argentina. And then I switched gears and I decided to leave the States. I actually went over to the UK and did a master's where I worked with Barbary macaques and studied stress and coping behaviors in a free-ranging population. And then I came back to the States and went and studied gelato monkeys in Ethiopia. These are guys that kind of look like baboons, but they're a lot prettier if you ask me. They've got really flowing hair. They've got a lot of cool both vocal signals but also visual signals. They have like a bleeding heart. That's why they're often called the bleeding heart baboon as well. And anyway, so these guys are really interesting in the lab or in the field. And then while I was there, I also got the privilege to start working with prairie voles and started getting bonded to them at that stage, where I realized they were just so interesting. And then postdoctoral work, yeah, I went back and I really loved marmosets, worked with them for a little bit, and realized I didn't really want to do primate work, at least in a laboratory setting. And so I went back and studied voles, and now here I am. Got to go over and keep studying prairie voles at Western. Okay, so coming back to this model of social bonding, so you can think about there being, I'm just going to talk about two general stages of bond formation and also bond expression, so kind of how you maintain and show those bonds across time. And we know that there are some really great hypothesized bonding networks in terms of areas in the brain. We've got regions that span from the prefrontal cortex all the way to the hindbrain, but also, and of course, also the subcortical regions as well. We're going to get into some of the nitty-gritty of these later, but I just want to bring it up that this is not new, this idea of there being multiple networks. What's been a little bit harder to do in the lab, especially in mammalian models, is trying to study all these networks at one time. It's quite common to cherry pick and focus on one region or one particular system, just because it's often been a lot easier to do than trying to look at the whole brain. Okay, so how do we actually study these hypothesized circuits in an animal model? We need a model that not only forms these attachments, but then has an opportunity to study them across time and how they actually are expressed in terms of selective preferences. And so one of the models that you can study for this is the prairie vole. If you're not familiar with the prairie vole, these guys are actually found in North America, and they do span up here into Canada, although unfortunately not here in Ontario. Most of the people who study prairie voles get them from Illinois and Kansas in the States. And these guys are known, they have a pretty well-established timeline for forming a partner preference. So if you, for example, the first thing you have to do is you actually tend to play Cupid, you pair two animals together. I'm not going to get into the details, but if you care about the reproductive side, females are actually induced ovulators. So in the lab, people actually use estradiol as a way to bring females into estrus. And then at that point, researchers will pair them together. And then within just one day of mating, they will actually form a selective preference. But what's really interesting is that there's variation there in terms of how strong that preference is and how temporary it is across the day. So if you just pair them together, they start to mate, a couple hours, let's say, they will not form a selective preference. If you pair them for 6 hours, they tend to form a very temporary preference where it'll eventually just go away across multiple days of testing. And then if you let them pair for a full day, they tend to have a very stable pair bond at that point, where they will actually stay bonded if you test them months later. And so it seems like this time course of 1 day of mating is really important for prairie voles. And then once they've established this bond, it's often tested by looking at their preference for their partner over a stranger. Okay, so what I want to do today is break up these two time points or these two general time points of forming a bond and expressing a bond. So I'm going to talk first about bond formation. I'm not going to talk about all the nitty-gritty of this side of the story because it has been published, but I want to set it up as a stage that will kind of also help to transition to the next part, which is work that we're still kind of grinding through some of the data with. So I'm going to talk first about this, what's this potential network that's coming out when these prairie voles form a bond, and then what is happening when they're actually expressing and forming and showing these selective preferences. Okay, so let's start here with formation. What we did was one of the questions here that's been really hard to answer in voles is whether or not these potential regions that are involved in pair bonding are just something to do with familiarity or comfort with other individuals. And so we designed a study where we're looking at essentially the time course of pair bonding. So when they first meet an individual, they have an unstable bond, and then they have a stable bond, but then also comparing those time points between animals that are male-female mating pairs, or animals that are cage mates and are familiar and already essentially have an established relationship, even if it's not like a pair bonding preference. And so we wanted to compare these groups to figure out differences across time, but also differences across these different types of pairs. And so we had a timeline that looked something like this, where we had some animals that would go through an isolation period of 2 hours, where they would just be in a chamber on either side of a divider. They wouldn't interact, at least from a tactile, in terms of like touch and stuff. So they would just be kind of alone on either side. Then we had a group where we would take the divider out, let them interact for up to a couple hours, and then and I'll tell you what we do after that. And then we have another group that was up to 6 hours, and then we had one group that would be paired for up to 24 hours. And we would collect a lot of data on them across these entire hours of socializing. And then within each of these time points, we had pairs that were male and female, unfamiliar, unrelated mating pairs, or we had animals that were essentially reunited with their cage mates. And so we had these same-sex siblings that are already being paired and already are used to each other in the colony. Okay, so what do we do beyond that? At each time point, we wanted to get a sense for what was happening in the brain. So we actually would sacrifice animals at each of these time points, and we would analyze their neural, I guess I'll get a little bit into that, but we would essentially look at CFOS expression in the brain. We would also collect behavioral data across time. So we did a lot of tracking across all these hours of interaction, but we also went through and had these 1-hour focal samples where we did a lot of measurement of a variety of different behaviors, including like grooming and investigation and huddling behavior. Okay, and so I'm going to talk a bit about the behavior first, and then I want to get into the neuro side. I'm not going to show everything here, so if you're really interested in the behavioral profiles, please come and talk to me because I have more data I can share with you guys and talk about. What I do want to point out is that if we take these different behaviors that occur in these focal samples, we get a general cluster of what we call inactive social behaviors and active social behaviors. So this is just a heat map here showing how the different behaviors cluster together. And so we get essentially, what I want to point out here is that we've got a group that is these investigation behaviors, but also mating behaviors. And then we get ones that are a bit more related to proximity. So things like approaches, but also following behavior as well. And then across these different groups, we tend to find that a lot of these active social behaviors are pretty high in these early stages of interaction. Not super surprising, but what's really interesting here is that we see, especially with mating behavior, you see a drop-off across time. So they're mating a lot in these early time points. So this is 0 hours, they're not actually paired yet, so we wouldn't really expect to see social behavior. But at 2.5 hours, they're spending a lot of time being social. So in this case, this is mating behaviors. And then at 6 hours, they're spending a little bit less time. And then by the 24 hours, they're pretty much just chilling in side-by-side contact. And then I'm going to talk a bit about the USVs. This is ultrasonic vocalizations. I'll talk a little bit about them towards the end of the talk. But essentially, they also are vocalizing a lot during these early time points in which they're forming a selective partner preference. So you're seeing these red bars because these are the ones that are mating pairs. So we see these, especially USVs, we see those and other active behaviors with those mating pairs. But we don't really see them with the same-sex siblings that are already familiar with each other. And then the other cluster are these inactive behaviors where are things like side-by-side contact, or the vole world likes to call it huddling, which is really just two animals kind of side-by-side just chilling. And for that, we tend to find that there aren't differences between mating pairs and same-sex sibling pairs, and if anything, it increases over time. So it's a bit of an opposite pattern, but what is really important to take away here is that we have pairs that even if they're not mating, they are spending a lot of time being social, and they are in proximity to somebody else. And so this helped us feel good about the fact that it's a good control of at least spending time with another individual. And so the sibling pairs will then act as a control for the mating pairs. Okay, so then what we did is all these 4 time points, we ended up taking brain tissue, and then we essentially did whole brain imaging of CFOS induction. CFOS is considered like an index of neural activity. And in this case, we're using this because it has worked well with prairie voles, but it's also been used pretty well with this whole brain imaging pipeline that I'll talk about here. And I do want to point out that we looked at whole brain imaging in a couple different ways. So one was to look at voxels. So there was, in the end, with we almost had 200 vole brains when you take everybody into account. We had 4 million voxels per vole brain and 864 regions that we're looking at throughout each of these brains as well. So a lot of data that we were getting to work with. So let me talk a little bit about this whole brain imaging pipeline. So part of the reason why we did this, we wanted to look at the whole brain, was because a lot of work, especially in voles, has been pretty targeted on specific regions at a time. And so we wanted to get a sense for, if we are just taking, if we're looking at the whole brain, what are the areas that are popping out as being super important at different time points throughout the pair bonding process, and that may or may not match with what has already been seen when people focus on one at a time. And so we used a pipeline. So before actually I started to postdoc, my advisor at the time, Steve Phelps, had connected with Pavel Austin, who was at Cold Spring Harbor at the time, and he had established this technique where he could clear a mouse brain, stain for CFOS, and then image the entire clear brain, and then use light sheet microscopy to basically pull out the cells counts across the entire brain, across those millions of voxels and different regions that were identified via an atlas. And so we were able to work with him and his team to take that pipeline and then transfer it over to and change it for the vole. So here's what it kind of looks like a little bit. So we have a reference brain where this top part here, we're seeing what the vole brain looks like compared to the mouse. In general, the vole brain is about 30% larger. They tend to be a little bit more stretched out than what you might see in a mouse brain. They actually have, fun side note, they actually have larger auditory cortexes relative to the rest of the brain compared to some other rodent species. So that might be part of the reason why. And then we also were able to parcellate different, we could use a reference atlas to essentially create boundaries for these 824 different regions. And so we were able to do that, and we're also able to look at voxels across the brain as well. So we had a lot of data coming out of this pipeline. Essentially, we would send our brains to Cold Spring Harbor, they'd run them through the pipeline, and then they would send all that data back to us, and then we would do a lot of the work on the back end. Okay, so essentially, just kind of summarize here, we have a common coordinate framework that is based on data from mice, but then we were able to apply that over to voles, and then use essentially light sheet focal microscopy to identify cell counts that were expressing CFOS at these different time periods. So it looks something a little bit like this, where we've got our CFOS staining, then we have the light sheet focal microscopy. Then we would use a pipeline or a computational pipeline to identify the cells that were active or not active. And we didn't, I won't get into that. If you have more questions, feel free to chat with me about it later. But essentially then we would have centroids for where the CFOS counts across voxels, and then we could parcellate these CFOS counts according to regions of interest. Or we had essentially all the regions from this Allen Brain Atlas. And then we were able to kind of take that and then essentially do a lot of analyses on the back end to see how this the CFOS expression changed across not only the time point, but also the two different groups of these mating pairs compared to their same-sex sibling controls. So it's a lot of data. How do you actually analyze it? we used a model approach where we would have essentially two models. One is like a hypothesized model and one is a null model. I'm not going to get into the weeds on this one, but essentially the hypothesized model would take into account not only the timeline and the identity of if it was male or female, but it would also take into account whether or not it was a mating pair versus a sibling pair. And so we were able to analyze not only these time points for differences between mating pairs and sibling pairs, but we also could look for potential sex differences. And I haven't really focused on that part, but it is a story within the vole world of trying to understand whether or not these differences in pair bonding, how much is related to sex differences. And in general, it's been a little bit of a mixed bag. And so we're able to use this as one way in which we could see if these networks are different between the sexes as well. So we had essentially this model that took into account kind of the basic information, and then this other model, which was more of like a pair bonding model, where we took into account whether or not animals were being paired with a mating partner or if they were just paired with same-sex siblings. And then we were able to take that, we ran it through essentially a post hoc correction, and then we were able to kind of look for the voxels. We first did the voxels, and then we looked at the regions of interest later. But if we're looking at the voxels that seem to be picking up on these differences of mating pairs versus the controls, we get something that looks like this, where we have, it's not that the whole brain is necessarily lighting up, and granted with our statistical approach, we're really kind of getting at the tip of the iceberg. So we're getting at the regions that are showing the most variation between mating pairs and the control. But essentially, how you read this is the front of the brain is over here, and then it keeps going, and the very back of the brain with cerebellum is down at the bottom over here. I just want to point out kind of a few key areas was that we found that some of the areas that were really predicting whether animals were with a mating pair, were within a mating pair or in a sibling group was in the prefrontal cortex, so that's up here. Another area that we saw a lot of potential variation was especially in regions of the bed nucleus of the stria terminalis and a lot of other hypothalamus regions that were close by. So that's this really bright area in A3. And I sort of skipped by this, but A2 was actually showing, I know it's kind of dark here, but it's showing the nucleus accumbens, which is an area known to be really important for reward and has been very well studied in the vole. We didn't actually find a lot of variation in CFOS expression in that part of the brain, which is something we're really grappling with. And I can talk to you guys about what we think is going on there, but in terms of this story, we actually didn't find it as coming out as a strong predictor. So then going back to here, we have the bed nucleus stria terminalis area, and then we had a lot of areas within ventral medial hypothalamus and amygdala off on the right. And so these are some of the areas that come out, at least when we're looking at all 4 million voxels across the entire brain. But it's a little bit hard to relate these voxels then to things like behavior, and so we ended up then focusing more so on specific regions of interest, or I guess not really regions of interest, but we wanted to look across the entire 824 regions that we were able to look at CFOS cell counts. So to then take the same approach, we also had a model comparison where we would have a hypothesized model versus a null model, and then within there we were actually able to do a bit more than that in terms of I used a kind of like a randomized, like a permutation approach where I actually randomized all the data to control for potential confounding variables. We're looking across a bunch of different regions. And so we're able to kind of add like a little extra layer there of certainty that we're finding areas that really do have significant differences between groups. And then what we get essentially was we found 68 different unique regions that seem to be good at predicting whether or not animals were in a mating pair versus a sibling pair. And I just want to point out a few structures here is that we found some regions within the isocortex. A lot of these were related to prefrontal cortex. Some other areas, is within, for example, the hypothalamus, which included a lot of ventral medial hypothalamus. And then we also saw a lot of bed nucleus stria terminalis as well. Okay. And if we take these then, so okay, this is great, but what it doesn't really tell us so much about kind of how those areas kind of fit together. So we did a little bit of clustering analysis where we wanted to see kind of how these different regions were kind of fitting together in terms of their expression. And so here we essentially found data that suggests that there was about 7 to 8 neuroanatomical clusters. And so these are regions that are, we think about it more in a functional way, of they're all kind of having the same signal. And so I want to point out one here that we call the BST cluster. It included bed nucleus of the stria terminalis, but it also did include some other regions, like postural, dorsal, preoptic area. So we kind of use labels that are kind of the candidate regions that were in there, but what's really important to note here is that these are actually pretty heterogeneous clusters. And if you look at how these clusters act across time, you basically get a little bit of different signatures across these different groups. So I just want to point out BST is the one where you get really strong changes, especially in these early time points of pair bonding. Okay, so that gave us a first look at essentially how is the brain responding to these, to the time course of pair bonding. But we next wanted to figure out what does that mean in terms of the behavioral side, because it doesn't really tell us much. What is it? Why do we care so much about BST or some of these other regions? What does it mean? But it's really hard when you've got 68 different regions and a bunch of different social behaviors. And so we used an approach called canonical correlation analysis to get at the dimensions of data, both from the side of these neural expression patterns, but also from the different behavioral metrics that we looked at. So it's a little bit like if you took PCA, but then you had two different groups of variables and then tried to find the dimensions of variation that fit in terms of combining those two sets of variables. And so by doing that, we were able to isolate the dimensions that had explain the most variation between groups. And if we plotted these, we could have basically kind of like PCA, like points, each individual would have a metric across these different dimensions. We find that the one that would explain the most variation was at these early time points of bonding. And so here, the red is representing mating pairs, blue is representing sibling pairs, and then the light are for female voles, and then the dark are for male voles. And what we essentially see is that we get a really strong pattern where you're getting just responses in the brain, but also these behavioral responses at both the 2.5 and 6 hour time points. So these are the areas of when they first start to mate, and then they form potentially like an unstable pair bond. And if we then tried to figure out what were the areas, both in terms of in the brain, that actually did the best at explaining this area of variation, or this dimension of variation, we found that BST was really the top area. So this is specifically the principal nucleus of the BNST. And this was the region that had explained the most variation across individuals. And if you look at the time course, so this is the 0 when they haven't been paired yet, early pairing, mid pairing where they have an unstable bond, and then 22 hours, a full day of pairing, they have a strong bond. We're seeing that this is the area that's having a lot of activity, especially in these early time points. So it's really matching onto what we see if we combine all the data together into this multi-dimensional data set. And then finally, one thing we thought this was really bizarre potentially at this point was, again, we were also looking a little bit for sex differences. It wasn't the main focus, but we did expect to see something that would pop out. And one thing that really caught our attention was that we actually see a lot of similarity when you look at males and females overall. So the light bars are females, the dark bars are males. So even at this early time point, they're almost at the same level. And same thing when you're looking at this later time point of 6 hours. So that led us to wonder, what's exactly happening there? And part of the way we can look at that is by looking at the behavior. So the BST is the area of the brain that had the best predictive value in terms of this canonical correlation model. The behavior that had the most predictive value was mating bouts, and these were specifically successful mating bouts. And so these were ones that essentially, this is showing the number of mating bouts across time, or per hour, and then how that relates to activity within the bed nucleus of the stria terminalis. And essentially, you're seeing a pretty strong correlation between how active the BNST is and their successful mating bouts. But the other bit to keep in mind here is that we're also seeing a similar response or similar relationship between brain and behavior, not just for males, which is on the left or on the right, but also for females. And we were kind of confused, like what's happening here? That we thought that we would see some sort of relationship with mating with males. It seemed to make a lot of sense. There's a lot of other research showing that BST is important for male-initiated mating, but we didn't really expect to see a relationship in the females. But it does suggest that the mating bouts are also potentially important for females, at least when you're predicting what's happening in the brain. And one thing I do want to point out here is that it seems like what's going on is that it's not that these two populations of males and females are showing overall the same responses. It's actually coming from coordination within the pairs. And so what this means is that if a male has a high level of brain activity, the female does as well. And so we looked at this in a few different ways. One is if you look at the multi-dimensional data set and you just plot this first dimension, you see a very strong relationship between what's happening in the female brain and what's happening in the male brain. So there's some sort of coordination there of, you know, whoever's having the strongest response within the BNST is also, for the males, is also for the female pair mate as well. And then you see the same thing, or a similar thing if you look at just individual brain areas like the BNST. And then if you were to plot, let's say, how the mating bouts are related to what's going on in the brain, you see a strong overlap between males and females. So this seems to suggest that, one, we've got at least areas of the brain like the BNST coming out as being really strong predictors of mating. But something we found just almost more exciting is the fact that the males and the females within a pair
are showing the same changes as well, or similar changes. So we're not finding
sex differences, at least in this parameter. We're actually seeing coordination between the pairs. And just to kind of broaden this out a little bit, we did look at some of the other candidate regions that came out of our analysis, and we looked at them across different time points. And so here, what I'm showing you is how the male and female pairs with their brains kind of correlate to when they haven't been paired at all. So you wouldn't really expect them to have any kind of relationship. This is just showing Pearson correlations where blue is a negative correlation, white is 0, and then warm colors are for positive correlations. And overall, if you took an average, this is pretty random. So here we're looking at the same brain regions, the 68 brain regions. We're looking at a heat map of how they relate between male and female brains. Then what we can do is look at what's happening at these early time points, 2.5 hours and 6 hours, when they're just starting to form a stable pair bond. In that case, you see quite a lot of positive correlation between the brains. So there's a lot of coordination. The pairs that have strong responses are happening both for the males and for the females. And then finally, we're also really curious to see, well, does this translate then to once they've actually technically formed a pair bond? And in that case, we see a much smaller cluster of regions that show a lot of coordination between the pairs. So here, I know you can't really see which particular regions they are. What I can say is that this is the amygdala cluster that we have. So we have a lot of, like, medial amygdala, for example, is within here, and that's where we see a focus of coordination between the males and the females within a mating pair. I don't have in here the slide for the sibling pairs, but what I do want to say is that if we were to look at sibling pairs, they also show pretty much no coordination early on. But then within these early time points and later time points, they look a lot like the bonded voles do. So you see a lot of coordination specifically in the amygdala regions. Okay, what time do I have until? All right, so that's all I want to talk about in terms of the pair bond formation story. I just want to point out that some of the key regions that seem to come out of this analysis were included bed nucleus stria terminalis, medial preoptic area, ventral medial hypothalamus, paraventricular hypothalamus, a variety of regions in thalamus and prefrontal cortex, amygdala, and I didn't get to talk about it, but actually auditory cortex came out as well. Okay, and then let's talk a little bit about pair bond expression. This story is still, we're working with a few pieces of it, so I am welcoming feedback. If you guys have ideas or thoughts on how we can do analyses, I very much welcome you to come and talk to me about that. So in this part of the project, we had a whole different set of animals. We essentially paired them together for a full day, so we didn't do any kind of manipulation of time course. We purely focused on males and female pairs. And then a day later, what we do is we separate them for a little while, and then we run what's called a partner preference test. In general, when people do this with prairie voles, they will have a partner tethered on one side of an apparatus, a male, and then a stranger tethered on the other side, and they see who the focal vole goes and spends time with. But we thought to ourselves, we wondered, one thing that hadn't really been done so much was dissecting that particular paradigm, because we don't really know, are they picking the partner because they find the stranger aversive? These paradigms are always a single stretch where they have to go to one end or the other. Or could it be that, they actually do prefer the partner and they're not just finding the stranger aversive? So we wanted to pick apart, what happens when they're just exposed to a partner, just exposed to a male, or put into a choice paradigm. And so we essentially had these different options, as well as an alone condition where they're just there's nobody else. But essentially we had a partner-only condition, a stranger-only condition, and a choice test or a partner preference test. Okay. And I wanted just to show you a little bit of what a partner preference test looks like. This is showing on a little bit higher speed. This is a focal animal here. And this animal is able to go through little doorways in these dividers. And on either side, there is a partner, which I think is on the left, and then the stranger's on the right. And they are tethered with a little fishing lure to the side of the chamber so that they can't move around, and only the focal can move around. And then below that, you're actually seeing the course of their movements based on tracking. And so from the tracking, you can see they spend a lot of time potentially over near the partner. And if we actually look at the data, we find this is just showing that for sure this is what we expect to see with a partner preference test, where if you look at what's happening for females and males, they're both preferring their partner over a stranger. And so this is a traditional partner preference test. This is what we want to see. We were also able to look at different behaviors. So we looked at things like where they're spending their time in terms of just the chamber side. You can also look at things like how much time they spend in proximity. And if you do kind of either approach, they show similar results. I'm not going to talk too much about this side, but one thing we thought was very interesting is that we see differences in the sexes when you look at whether or not they're just exposed to a female or a partner or exposed to a stranger. And so here what you're seeing is how much time they spend near their partner when they're just exposed to the partner, or how much time they're spending by a stranger if they only have a stranger available. And this is showing two different types of behavior, whether or not they're spending the time in the zone, or whether or not they're in active or in proximity or huddling. And one thing I want to point out here is that we tend to find that females overall are still spending a bit more time with their partners than females that are just paired with strangers. But you see variation with males. So actually males who, if their partner's there, they spend time with their partner. But if they are with just a stranger, they still spend a lot of time with the stranger. So even if we do see differences in neural expression, it might be due to these differences in behavioral strategies of, males might be still preferring to spend time with their partner, but if there's a stranger there, they're going to investigate. And so we do see these differences in terms of between the two sexes. And you can also track what they're doing across time. And all I really want to point out here is that they tend to kind of settle around 100 minutes or so, and then they usually will just stay there and huddle, and then they are there for the rest of the time. But you do see that females and males are both tending to spend a bit more time by partners, but males especially will also spend time by strangers. I know it's a little bit hard to read there, but these light blue lines at the bottom are the females that are being exposed to just a stranger. So they're not really spending much time at all near a stranger. Okay, and then as part of this study, we also then took out the brains and did the same kind of CFOS whole brain imaging that we did with the first study. So we did, again, these light sheet focal microscopy based off of stained CFOS brains, then did segmentation to find the voxels, as well as also the different brain regions where we had CFOS cell counts within there. And then we used, again, a model approach where essentially in this case, we're comparing a null model, we're just putting sex in there and the block of the study, and then the hypothesized model is predicting social conditions like being exposed to a partner, being exposed to a stranger, or being exposed to a choice condition. So ideally, we wanted to find areas where they were reacting to a sort of social paradigm. And what we get is a lot less of activity, or at least a lot clear variation across brains when we use a voxel-based approach. But what I do want to point out is that we do see that some of the regions that are expressing variation between males and females that are being exposed to their partner versus a control condition, or a partner stranger versus a control condition, are in sort of near the prefrontal cortex. We also, again, see the bed nucleus stria terminalis showing up. We also see ventral medial hypothalamus, and funny enough, we also see some regions in the back in the midbrain here. And essentially, we can also do the same thing then to the different regions of interest or across the entire brain. So again, we have this model comparison approach. And if we do that, we see a selection of about 20 regions that pop up as being above our criteria. And what I want to point out here is that just like in the first study, we saw BNST, bed nucleus terminalis, as being pretty much an outlier in terms of doing a good job predicting which social condition it was. But we also see some new areas like entorhinal cortex, that's what ENT is here. And we also see auditory cortex popping up again in this story as well. So we see some similarities, but also some potential differences here. All right, and I'm going to speed through this one here, but we also did the same sort of thing. We looked for clusters of different behaviors. We got essentially a cluster of BNST-related regions, but we also saw other regions related to memory areas like the entorhinal cortex, and also this is tubomammillary area as well. Okay, so just to wrap up that story quickly, what I want to say is that in general, we tend to find some overlap between what's happening in the brain during pair bond formation and pair bond expression, especially when we're looking at the bed nucleus terminalis, parts of the hypothalamus, and also auditory cortex as well. But we also see some differences, specifically that the entorhinal cortex, which is a structure in the brain thought to be associated with memory, seems to come out as predicting groups better, or predicts groups in this expression stage more so than in the formation stage. So, taken together, what we've been really excited about is this really does a good job of bridging a variety of different networks, both from human and animal research. So we've got some really classic work looking at parts of the BNST and preoptic area in male sexual behavior, and this has come out pretty strongly, at least in this study here. We also see areas related to social reward, like preoptic area, and parts of amygdala is coming out as also being pretty key here. And in terms of Preval's work, there's not been quite as much focus on amygdala, so this is pretty important here. Also, a lot of regions thought to be important in human attachment, especially prefrontal regions, and again, social rewarding areas as well seem to be supported with these data sets. But then also supports this network of pair bonding that, even though historically has been focused on individual regions, we have shown at least a good chunk of this network comes out when we look at every area together. And then just to kind of, then I want to spend a little bit of time talking about what we're working on now. So a lot of this is kind of work that's already happened, but it's really set the stage for where the lab is going right now. So we actually just got prairie voles. We've established a colony over the past year. It took us a while to get the animals. And then this past year, we finally got them at a pretty stable stage. And so we're just starting to collect a lot of data, or starting to collect data with them. And one thing that we're very interested in is in the communication side of the pair bonding story. So I've talked a lot about what's happening from a physiological standpoint, but what I'm very interested in is what's happening in between individuals. How are they actually exchanging communication or exchanging information in between each other? What role does that play at different stages of forming an attachment? And so we're focused a lot on this angle of the story, but then approaching it from different biological perspectives. And one thing about the voles that is actually was one reason why I got into them is that they, even though you can't tell because they vocalize in the ultrasonic range, they actually have a pretty diverse vocal repertoire. This is very similar to what you see in mice, but also in rats. And what we have essentially done is to look at how this has changed both during the, or how they vocalize during the formation of bonds and also in the expression of bonds. And so this is tracking from pairs that have been bonded for a full 22 hours. This is 22 hours of vocalization data, where we were able to pinpoint the darker areas are where there's high vocalization rates. The top is showing mating pairs, and the bottom are sibling pairs. And so we see a lot of vocalization, but it's happening across the entire day of pair bonding. And this is just an aggregate measure down here, where we're seeing the red is the mating pairs and the blue is sibling. And yeah, essentially the mating pairs are chatting a lot. So we get a lot of vocalizations, and this isn't just immediate. This happens even after they've been paired for a full 22, 24 hours. And then we also have analyzed a lot of data from when they're forming or essentially expressing pair bonds. So when they're in this dynamic of being in a choice situation where they're picking between a partner and a stranger, and when they're just being paired with a partner or just being paired with a stranger. And this is some work that one of the grad students in the lab has taken on from some of the work from UT Austin and is now following up on in the lab, where she was able to show that the total rate of USVs is, as you'd expect, higher when animals are paired with somebody than when they're alone. So this is their USV rates or their vocalization rates when they're alone. So a lot more vocal when there's others, but then there's a lot of variation in terms of when they're vocal. So when they're with a partner or when they're making decisions between a partner and a stranger, they have, they're vocalizing, maybe like 3,000, maybe like 1,000 times an hour. When they're paired with just a stranger, they're actually very vocal. And this is a story that we're still trying to figure out because we did not really expect to see a high rate of vocalization when they're being paired with someone they don't know. Could be related to maybe alternative mating opportunities, and that is something that we would like to follow up on. And then the other thing that we're working on is looking at particular or potential regions in the brain that might be helpful for producing vocalization at different stages of the bonding process. And so there's another graduate student in the lab who has essentially done ibotenic acid lesions compared to controls like saline injections and sham animals. She's focused on the prefrontal cortex, and then after these guys have essentially recovered, then they are paired. These are all focused on males. The males are then paired with a female, and then we do these acoustic interaction tests across 3 days of pairing. And we're able to essentially say what's happening with the male versus the female. And we're finding some interesting data that in the past people have assumed that most of the males are vocal, and generally we find that, but we also find that females are vocalizing a lot too. It's not just the males. And then this is showing what this looks like when they're actually in these chambers. And she's done some really cool tracking work showing that the males are spending a lot of time oriented towards the females. And so it's not just that they're vocalizing randomly, it does seem like they're vocalizing in the direction of where you'd expect them to vocalize. And then what she does is she does partner preference tests at the end to see what the males are doing. And so here he has his female partner on one side, female on the other. And so we're able to now figure out if males that have damage to the prefrontal cortex are showing different preferences here, but we're also slowly getting through some of our audio data as well to see if they have, there's different levels of vocalization happening while they're doing this paradigm. Okay, and I just have some preliminary data I want to show you, but we are seeing some potential variation in how males are producing vocalizations based on whether or not they've had damage to the prefrontal cortex. So this is an example from a control animal where they're producing what we really expect to see. These look like courtship vocalizations on the spectrogram here. And then this is an example from an ibotenic acid lesion animal where they have vocalizations, but it might not be at quite the same rate, perhaps also not quite the same bandwidth and frequency modulation. But this is definitely still a work in progress. I think the students have just gotten through analyzing a big chunk of the data. So this is very tentative, but we are really excited to see what will happen in terms of, is the prefrontal cortex damage resulting in variation not only in the rate at which they're calling, but also in the structure at which they're producing these vocalizations, and then does that also track with how the males are making decisions when they're presented with their female mate versus a stranger. And then exciting work too is that we have one of the other graduate students is taking some of these recordings from both the lesion animals and the saline animals, and we're hoping to present them to novel females to see if the females treat them as differently as well. And so we can see if that leads to repercussions from a fitness standpoint of actually being selected for, having males be selected for by females. Okay, so with that, thank you guys so much, and I definitely ran a little bit over. I just want to also thank, a lot of this work I presented today was in collaboration with Phelps Lab and Austin Lab at Cold Spring Harbor. And then the Gustison Lab team is just starting to grow now, but it's been a real honor to work with everybody so far. They've just been so excited, and I'm really excited to see where everyone's data and projects go next. So thank you so much for listening. I'm happy to take some questions.
Thank you for a really interesting talk, Morgan. I wonder if you could just comment for a couple of minutes on why a stronger or
Yeah, it's a great,
What do we know about that? And related to that, I'm kind of really confused, but I probably just didn't hear it right. Why is it important to form the pair bond after mating? Why doesn't it happen before mating? So those are two kind of more evolutionary or behavioral
yeah.
questions.
Okay, so for the first question, sorry, Megan, it was,
What does it buy you? The strong or weak pair bond? How does it affect your fitness?
yeah. So one thing I can say is in the field, there's been some really cool work where folks have looked to see, specifically in males, because in the field, females will tend to have pretty established territories, and you get pretty separate territories across space. The males, there tends to be different strategies on how they deal with that. So some males tend to actually go and what we presume is form a partner preference with females. And actually how these guys were found was people were out in the field collecting, doing traps, and then they kept finding the same animals being trapped at the same time. So that would be an example of a resident male. And then I'll come back to that. The other strategy people have found is like a wanderer male, where he'll actually go around and kind of, almost like a sneaker male, try to get matings. And they're socially monogamous, not genetically monogamous, so this does happen. And it seems like what tends to maybe be the case is that there are alternate strategies that are both essentially great for the male. So folks have looked at the number of offspring, and they're actually pretty comparable. So it seems like, well, we might be also, so this is where the diversity is really key, and in the lab, I think just because of how we're pairing animals, we're probably somewhat artificially selecting for the bonded male. But it is interesting to note that in the field, it is like one of the strategies that seems to work well for them. What was the second question about?
Why would the pair bond form?
Oh, after.
rather
Yeah, that's a really good question. So I don't know if I have a specific answer to that. Just from the literature in voles, we just know that happens. Like they have to mate first, and then they don't mate. But if they're in the same container, but they're separated, they don't form a partner preference. I wonder, like maybe it is maybe a compatibility kind of issue where they gotta, show some sort of preference or so that they can mate. It is one thing that makes me think of is I've always wanted to look more so at how they make those decisions. A lot of the vole work is kind of, we play Cupid and we kind of jump past the mate choice part where we just say, okay, you two are just gonna be paired together. And oftentimes people use estradiol as a way to bring females into estrus and just jumpstart the mating process. In our lab, I'm trying to avoid that. I want to have a more naturalistic formation of a pair bond, which definitely is just going to take longer. What is maybe kind of interesting here is that we don't know if the mating is somewhat related to decisions that they make. And what I mean by that is that there's at least one study where they had animals exposed to each other before they would do a mating. And so it takes a while for the females to go into estrus. It's at least a couple days if they're not being hormonally induced. They find that if they pair females with males that they somewhat showed a slight preference to before they've been able to mate, they will actually form a stronger partnership later. And how they did measure that is just like how much time they spend in the partner with a stranger. So I do wonder if maybe that fits a little bit into the compatibility hypothesis, but I'd actually be really curious to hear what you think about that, because I'm not sure. Yeah.
Could there be a relation, sort of building on Stella's question, between pair bonding and like offspring raising? Like if a bonded pair could be like better at raising a child potentially, like a full kid?
Yeah, so it is better to have a pair. Offspring do better when there's biparental care. Yeah.
Would that be maybe a factor?
So that would definitely, like if nobody did it, then probably the offspring survivability would be decreased, yeah. But from the wanderer male side of the story, they probably want to have another male sort of take care of their kids, yeah. Yeah, that's a good explanation. Yeah.
So the sea frogs usually use netting. And what are you connecting to the other stories about Gustison, many other neuromodulators? I wonder whether the netting can bring you to another level of understanding the changes that occur at the neuromodulator level.
Yeah, and I think this is where, so some of what we were a little bit surprised that BNST came out as so strong, and we've had some conversations with folks that have focused more on that region, and it sounds like actually the oxytocin system is strongly focused in that region of the brain. So it does seem to support this oxytocin idea, which, yeah, the oxytocin system is thought to be really important with pair bonding, although I do want to point out that there are some really incredible researchers out in California who have actually done knockouts of voles and oxytocin, and surprisingly enough, the oxytocin knockout voles still form partner preferences. So it's maybe more complicated than we think. Yeah. But yeah, that's a great, and I think this is where, so I haven't been able to do any follow-ups with this approach. It would be really nice, though, if we could focus in on, specifically oxytocin neurons. And this would be something where it'd be a good collaboration with people who maybe could do that work. Or like, if we wanted to try to do like a stain afterwards, although it just wasn't possible because the tissue wasn't, we weren't able to work with it after the pipeline. Yeah. Yeah, yeah. Yeah, they've done a little bit with tyrosine hydroxylase, but just on a general level of where is it expressed. We haven't mapped the two. But yeah, it would be nice to do, if we did like a whole brain approach and then try to match, that would be really, really cool. Yeah. Any other questions?